Hi, this is Dr. Ben Morrill. Welcome to episode 56. I don't remember.
Yeah, 56.
56 of Reptile Genetics Weekly. And glad to be here live. And, we have been preparing and doing all kinds of stuff these last couple of months. And today is no different. Lots of stuff going on today. So we forgot to talk about which episode it is. Kayla always has to remind me, but you guys get to see that live this time.
I got you, Ben.
So as always, not many people here yet, but with all the videos, we'll start out with letting you know about results. And so because we've been going through a pile of research data, I don't have everything to Sean like I usually would after a panel run, and the Fassst testing, Allie, that you'll meet here in a couple of minutes. The Fassst testing that she did today, I don't have that analyzed yet either. So we did get sex determination results done for colubrids and ball pythons, and then either late tonight or sometime tomorrow, we'll get the Fassst test results that I should have had done earlier today. And then probably sometime tomorrow or Thursday morning, we should be able to get to all of the panel results. So that's the update for that. And welcome Kayla and Sean and Allie this time.
Hi, it's way busier on here than
normal. What's up, everybody?
Yes.
Yes, and they finally get to see Allie's face. Well, okay, actually, if you went to Arlington NARBC, not this year, but the previous September, you got to see Allie when we were both bopping around there. But otherwise, it's our secret weapon.
And this time, everybody, even people on Instagram get to see. So
yes, you hit the go live on Instagram.
It's going
perfect. So we won't be able to see everybody's Instagram comments on this. That's a limitation on StreamYard. So Ben, if you can glance at that every once in a while, and if there's a super cool question that you want to do, then tell us.
All right.
Otherwise, we can see all the other comments on YouTube and Facebook.
Sounds good.
Yeah, and we got people coming in. I see lots of familiar faces.
Look at everybody.
Oh, I got to not cover the camera when I'm scrolling here.
Oh, yeah, that's okay.
Questions coming in already.
Good. Yeah. Let's see, Allie and Sean, you should be able to put a star next to those comments if you any questions that you don't want us to miss.
Okie dokie.
There we go.
Thanks everybody for tuning in. If you guys wouldn't mind maybe doing a shout out on some of your favorite Facebook groups or things like that, to let everybody know we're live. We usually do this for about an hour to 2 hours, depending on how much BSing and fun we're having.
Oh, we've already been doing a little bit before the stream started, so it was good.
Yeah, we've already been at this for almost an hour, so we're already being pretty silly. So yeah, let everybody know, please.
What'd you say, Sean?
I said, so yeah, let everybody know, please.
Yes,
that
please.
We're out there.
Give a holler on World of Ball Python's Facebook page or something. Story. While we're waiting for people to come in, the story that I was telling you three that now you guys are here. For those of you who don't know, I started for my other job, because this one's part-time, but my other job is at a wildlife rescue in the area. And we were talking about how turtles are constantly hiding their power level. In such case, we got a big snapping turtle that was this big around in the center. Just a common snapper, feisty, ferocious. It was still cold out, so they come in a lot. We had him back in a sink in the x-ray room because as you do. And I went back there one morning to check on him, and I looked in the sink, and no snapping turtle. Where did it go? And then I look next to my foot, and there's old boy backed up into a corner looking up at me, right next to my foot, and he's just like, "Hey, how's it going?"
Thanks.
I'm like, "Oh,
awesome!"
He had a 3-foot drop. There was a platform for him to sit on in the sink that we were keeping him in. That was full of water and everything. It's his hospital tank, and he managed to get himself up probably about this far up out of the tank, and then flipped himself onto the floor. Man, got away with no injuries or anything, but anyway, we put a grate over the top of that. Oh my gosh. Turtles are hiding their power level.
Yeah, and they can
Okay.
really snap down on you too.
Oh yes. Did learn that actually we humans have a stronger bite force than common snappers, not alligator snappers. Those things are a different beast entirely. But
Yeesh.
yeah.
All right, so
Okay.
Allie's afraid to say anything, so I'm gonna make Allie speak up. Well, I know she had some other stuff she needed to go do tonight, so Allie is our secret weapon, as you can see. She's the person that if she didn't prep the shed, then I did, and she runs most of the tests now. And she is now our second full-time employee at RGI. That's Dr. Ben. So she is cranking every day in the lab, turning out the test results. So all those fast tests that you guys have been getting, those results are coming from her work. She receives them, gets them prepped, and runs them all pretty much on her own. She's the boss. You want to tell, huh?
Said I do my best.
You want to tell everybody a little bit about yourself, since they hear about you all the time, but you never actually get to be on any of the shows?
What about me?
I don't know.
You're awesome.
There you go. I'm awesome. Kayla's words. No, I don't really have much to say.
That's okay.
I'm
Okay,
I'm
so-
with you.
Snake person like these guys. I like working with them, but not owning them, I guess. But
Yeah.
other than that, not much to say.
Okay, she's not a reptile person, but she's actually one of the best employees that we've ever had in my other companies. So she rolled over from my other companies where she's worked for me for what, going on 3 years now, right?
Mm-hmm.
Yeah, and she was killing it over there. And because she did so well, I offered an opportunity to start training for RGI, and now she's running the place. She mostly just tells me to get out of the way, which is great.
Yeah, I do that. Yeah.
Often.
Like if that's any indication of how good Allie is, like Sean is our money guy, and not anybody can just tell the money guy around. Allie does. She's very important.
Pretty much bosses me around every day.
Of it, okay? 3 years.
We work together in the office and in the lab every single day. So yeah, and we have for a few years now.
There you go.
Yeah. Okay. So Troy already asked me a question. This is my basement, Troy. I am that guy, the dirty old man who lives in his basement. That's me. So yeah, that's my man cave. You don't get to see all the cool stuff to my left, but it's all my gaming stuff and my books and all that stuff. So
the books are what you have hidden, though.
Yeah, I've got lots and lots of books.
Is that a chair with tennis balls?
What?
Is that a chair with tennis balls?
Behind you, Sean. Behind you, Sean.
Tennis balls.
I don't see. Oh, no. They look like tennis balls, but they're just little.
They kind of do, yeah.
Pieces.
Hang on, let me like blow up the image there a little.
Those actually used to be in the office that Allie and I worked at, but now they live in my basement.
Okay, it's just fancy. Okay. Anyway.
Okay, Troy.
So do we want to talk business?
A little bit at a time. Let's have some fun, right? I mean, but yeah,
Sounds
we've got a few interesting things to drop. Ben, you want to go first?
Yeah, man.
First of all, from Instagram, we've got a couple of things. Feely's Clutch says the person on the top left is questionable. So sorry, man.
Oh,
Let's
it's either
see.
Ben or Sean. I've swapped them around a little bit.
No, that was me. Yeah, sorry, John. I'll try to. You had to spend a night in the hotel around me. You know how questionable I am. So, let's see. Monster Ranch Exotics and Serpent Labs. We're talking a little bit about Mackenzie and Zebra. We have started working on that, but we don't have anything yet. So we'll have to keep wondering about that one. Any other comments you want to bring up from? We have some of them starred.
I think.
Or do you want to go to the first slide?
Soon. how long for monsoon, and Canova spilled some hints that there's some genetic compatibility for Xanthics.
Yeah. So both Monsoon and Monarch, we know the general area they're in, and we have a test that works really well, but not good enough to release yet. So Monarch and Monsoon are definitely coming soon. One of the cool, interesting things we can tell you about Monarch is that it is on a different chromosome than Ultramel. And so they are different mutations on different chromosomes. So they should not be compatible from what we're seeing. They're in completely different spots. I know some people have wondered about that. So, and there's a comment, someone was talking about MJ, Xanthic, and GCR.
Yes.
Yeah, there we go. Me waiting for GCR to be the same as MJ.
So that's a good name, Prophecy. That's a good prophecy. So what we've seen so far, and it's only a few, kind of like what we said with Trick, Hurricane, and Blitz. When we tested a few Blitz at first, we're like, yeah, it's identical on the few we tested, but you never know if someone, mixed up what line they had or whatever. So the few MJs we've run are identical to GCR so far. But I wouldn't say we're necessarily completely sure about that yet. And that GCR test, which so far is also working for MJ, that test looks really good. We had 2. So like we've said, we usually run at least 100. We had 2 that were reported as being homozygous or visual for GCR, but the test said they were hets. So I'm having to chase those down and see if, I'll have those people send those sheds again, and just maybe there's a shed mix up on my end or their end. So that test may be ready in just a week, or if we have to play with it more, it might still be 2 or 3 or 4 weeks, or, as those of you that have followed for a while, if I have to go all the way back to the beginning, then could be, 4 months out again.
It's like that sometimes.
It looks really good. And it wouldn't surprise me at all if we get those 2 back and they end up being clean. But if not, then we've got some other ones we can try and clean it up to last a little bit. That puts us at like 98% accurate. We want to be 99 or above. So it barely doesn't pass.
Makes sense. So it's a hard maybe on those 2 being the same.
Yeah. So I've tested, I think, 5 or 6 from 2 different keepers, if I remember right. But I want to try at least another dozen from at least 2 or 3 different keepers. I do have some MJs. Give me nope, I don't have his. I think his last name is Sukup. But anyway, there was somebody that sent me some where they had the information about who they imported from in Canada. So they're, have some lineage information directly to Canada as being MJ. So if I test those and those also are identical, then I'll feel really confident saying that.
Yeah, sounds good. Colorado Cold Blood says they appreciate how quickly the sunset test process.
So Allie, that's for you.
Yeah. Oh, thank you.
Well done.
I do my best.
Yes.
Northwest
that's Scott.
Says hello.
Thank you, Scott. I've been talking actually before we started doing any ball python morph testing. Scott made a large order with me, ordering ball pythons, and it was just as we were going to start doing a lot of this testing. And I let him know, hey, I really appreciate this order, and just so you know, kind of sounds crazy, but I'm going to try to do a reptile genetic testing business, and all this money is going to go right into that. So thank you, Scott. And he's stayed in touch since then. And we've run several tests for him. Actually, part of the data I have, I've got data at least from one animal I know that's GHI Mojave combo that I sent, or GHI pastel combo. I can't remember for sure, but he was POS OD and POS et Pied. So I'll let you know, Scott, in the next day or so if he's positive for either or both of those.
All right. Hey, look, it's Sam Ceresi. Hi.
Hey.
this guy is the youngest ball python breeder we know. We did an episode with him and his family. Great family.
I think Xander's the son.
Oh, that's right. Okay.
Yeah.
So Xander's, yeah, that's right. Xander's the son, and Sam Saresi, I guess, is his dad.
Yes.
All right. See, we got a message about a frozen stream being frozen. Everything looking okay on y'all's end?
Looks good to me. I can see everybody.
Yeah, I can see everybody here.
Having a problem. It looks like it's moving on Instagram too, so
that's good. All right. Let's see. Any news on Black Axanthic?
Not yet. We'll definitely be looking. So we've got lots going on with Axanthic stuff, and not only just ball pythons. We've got Axanthic in black-headed pythons, and carpet pythons, and retics. We've got it in several different things. But yeah, we will be looking at black Axanthic as well.
The hope is if we get them all nailed down in ball pythons, it'll help us nail them down in some of the other subspecies, and we can start offering them there too.
That would be cool.
And we have started getting more black Axanthic sheds in from people. So thank you. Those of you that have watched the show and sent sheds in, we appreciate it. And that's something that Canova is interested in. And they did a post earlier today. They'll be making sure we get enough sheds to figure that out too. So
yeah,
Axanthic is on our minds. That's for sure.
Oh yeah.
Have we even talked about TSK yet?
Nope.
Nope.
So that's the one that is available. So it's ready as a fast test already. And it will probably be, if it's not already on Morph Market, it probably will be tomorrow. Would you think, Sean?
Tomorrow or the next day at the latest. Yeah.
Tomorrow or the next day. It
I literally warned them yesterday and then confirmed today that it is ready to go. So
I think Clutch said pretty much the same thing. They'll have it tomorrow or the next day. So
yep.
Yeah. So yeah, TSK is good to go. It's very clean, looks great. Want to thank those of you that sent in TSK sheds. There's a lot of people that sent them in, and we had a lot to work with. That it took an extra time than I thought it would of going through all the sequencing, but it looks really good now. And the fast test is beautiful too, so it's all good.
And a couple people are asking about Paint and Sentinel.
Yep.
So Paint and Sentinel is coming very soon. We got a huge amount of information, a whole bunch of hits. Paint and Sentinel, I'm going to be designing and ordering. Also Acid and Confusion. There's 5 or 6 more that I'm going to be designing and ordering. And then in another week and a half, when Allie and Sean send all the panels to me, I'll be able to do all the panels, but then also on that, I'll do a research run and try some of these hits for Paint Sentinel, for Blackhead's another one we've got. So yeah, there's several cool ones that hopefully in a couple weeks I'll have some updated information on those, but they are coming very soon. I have good hits, but I just have so many that I can go after right now that it's going to take probably a good 6 weeks to get through everything. And we're hoping we have, 10 to 15 new tests over the next month. So.
Fair enough. What's up, Sean?
So just, I want to jump in on Ben's talk here.
Yeah.
I don't know if it was the last live that I did with you guys, which was quite a few months ago, or maybe the one before that, because I think we've only done, this is the 3rd or 4th now. But we asked all you guys, hey, what would you rather us do faster tests or more tests or whatever? And we continue to try to work on both. But the reality is we spent the last 4.5 months rolling everything over to fast testing, so you can get those results within a couple of business days. And now it's down to 1 business day. The good news is now that we've got that rolled out, Allie's working full time, smashing things in the lab, doing a fantastic job. Then now we're focused on research, and today's live is about that. We're turning over all of the sheds that you guys have been sending in and have given us mutation information on. It's all being sequenced at a higher level than we normally test at. And I mean, in this first couple of batches of information that we've gotten, we've managed to kick out as many tests as we have. But this last batch has given us a potential, we've already hit TSK. There's probably another 15 to 20 more in the basket. that we're working on, and that we have another run scheduled for probably 2 weeks from now, where we're going to have another 300 sheds being analyzed across another 10 to 15 mutations, and some of those will be duplicates because we want more information on genes that we're working on now, but some of them will be new genes like paint, sentinel, or things like that maybe we couldn't nail this time. So fast testing is over. Everything's working really clean fast testing wise, and now we're full bore research. We're turning over all kinds of stuff and getting all kinds of neat things back. There are a lot of things we're talking about today. There's a lot of things that we're not going to talk about on purpose because we just don't know enough, but we have some stuff that we suspect that's pretty cool. So anyway, you'll be
another pretty cool one that's interesting to talk about right now with monsoon. Like I said, and those of you that are just coming in, I see there's still monsoon questions. So Kayla might have to put that on the repeating thing. The monsoon test is very close. We know pretty much where it is, and we just have to find a test that works 100%. The monsoon and the monarch test right now, it's like 75% accurate. I don't know if there's a second mutation or if we just need to find another hot spot. But anyway, the interesting thing with monsoon is that it's very close to albino, so just the normal albino. And that's interesting. Those of you that watched our back to school, we talked a little bit about the banana and coral globe mutation. And so because banana/coral globe, since that mutation is so close to the sex determining region on the sex chromosome, that's why if you have a male that's a male maker, that banana mutation is on an X chromosome or a Y chromosome. So 90% of the time it stays with it. There's no crossing over in between. So 90% of the time it's going to make male bananas, but every once in a while there'll be a crossover event in between. And that's when the 1 in 10 bananas that he'll produce that will be a female. Well, because monsoon and albino are as close as they are, and different species and different chromosomes recombine at different rates. So I don't know, but just a heads up to any of you thinking about monsoon and albino, making an albino monsoon, that may be more difficult. It might be imperceptible. Maybe they're far enough apart and there's enough recombination near there. We won't notice it, but it could be similar to banana where it's like 90% of the time, it's not going to give you both albino and monsoon on that same chromosome. You have to get them both on the same chromosome twice to be able to make an actual double visual. So just a heads up, that might be a little more complicated than we thought before.
Yeah, to be clear, that's our going theory right now because they're so close to each other. But don't run out to the world and tell everybody for sure yet. However, they're really close. really, really close.
Yeah, end that sentence with a star and, like a little asterisk and say, as far as we know, so far. By the way, Ben, did you want to explain sorry if y'all heard the thump. My upstairs neighbors we believe they're werewolves. But you sent a really cool picture to me that's the thumbnail for our live stream. Did you want to talk about that? Because we were talking about axanthic stuff earlier.
I just thought that was kind of an interesting portrayal of, some of the genetic testing that we do. So there, the way this kind of a test works, you actually and this is the same as with yellow belly, the test that Dr. Siddell set up. So there are things called restriction enzymes, and they're proteins that will go and cut at a specific sequence. And so you can use both PCR and restriction enzymes to cut, and it can give you differential banding depending on the sequence. So with yellow belly, that works. And I think that Dr. Siddell had a couple others. So this is just kind of a portrayal of that type of a genetic test so that y'all can see that. It's kind of fun. Maybe too many bands for some people, but at least you can see what the two homozygous ones look like versus the hets and the negative there. So
Yeah, so this is part of what you're reading whenever we're doing our Fassst tests for certain genes.
Sorry, say that again.
Oh, sorry. I was saying, so this is part of what you're looking at when you're reading, like when you're getting the Fassst tests in, and then the other tests for some of them.
It is an example of one way that you can do that. It is since you can do a PCR and run it on a gel, then it's a pretty fast way to do it.
That's cool.
Yep.
Yeah. I think we got a little update on turnaround times. You might have seen this on our social media as well. Sean was talking about how, let's see, fast testing should, in most cases, should be next day. Ben, did you want to talk about did you want to go through these, or what were you thinking? If it's easy for you to see here.
Maybe even have Sean or Allie talk about this, because that's on their side, a lot of that.
Oh, yeah.
Allie, you want to run with it?
Run with what?
What's on
We're working on the Fassst test. They'll be 24 to 48 hours, right? Preferably,
yeah, we're trying to 24 now. So.
We shoot for 24. And then the panels is every 2 to 3 weeks, and the sex determination anywhere from, I'd say, 2 to 5 days. Yeah.
That's cool.
The reason the panel wait time is reduced a little bit is in slow season, we only do one sequencing run a month. But when it's busy and we have more sheds coming in, we're doing it every other week. So it typically takes us a couple of days to get everything prepped. And then when Ben gets the stuff, it takes him a couple of days to do all of the purification and library prep. And then it goes off for sequencing. And then when it comes back, it's another day or two of data analysis. So if we have your sheds within a week of the sequencing date, you should get results about a week later, 4 or 5 business days from the date that we actually run the test. So yeah, that's pretty cool. But yeah, the Fassst test is what I'm excited about.
Yeah.
Fassst test is, we're trying to knock it down to one business day for just about everything.
Mm-hmm.
So from last week, what's different is we do have Enchi and Orange Dream working, and then also TSK. Those of you that are coming in late, that's the new one, the test that works, and it is working as a Fassst test already. So yeah, we have everything. The only one that is taking me an extra week or two is Russo, but everything else is going. So Monsoon, Monarch, GCR Xanthic, MJ Xanthic, those are the ones that are likely going to be the next ones that will be tests we'll have available. And then, as specifically as Fassst tests, we're going to try to have them available immediately as Fassst tests. Every once in a while, we might make it available, and then it might take another couple of weeks for it to be a Fassst test, but we're going to try to make that immediately available as much as we can.
Awesome. Let's see. Sean, you want to talk about the pricing changes?
Okay. See that because they just want to hear all of the information about mutations, but we have run thousands of you guys' sheds now, and we appreciate the business, and we promised you guys last year we would start moving on some pricing. So that's what we're doing. Most of the tests that we have lots and lots of experience with now, we've lowered to $50. There's a few that are going to stay up there a little bit. Enchi, Red Stripe, Leopard, Orange Dream, Black Pastel, Sunset. Those are all kind of newer tests. But everything else on the list, and TSK probably will be on the short list too. But everything else is now Fassst testable, and it's $50 or less. We have a few single gene that are actually as cheap as $35. So
awesome.
Yeah, pretty cool. It's kind of a thank you to you guys. And honestly, like once we feel really confident about a test, it makes sense to lower the price because we know the test is working really well. There's not any problems with it. And there's no reason that we need to continue to kind of charge the research pricing for that test. So the other more expensive stuff, we charge a little more because sometimes we have to go back and tweak them, right? That takes more time and more money. So we always charge a little bit more on the newer tests. So anyway, $50 or less for 90% of our tests. So yay for that. And that's all live right now on Morph Market. I think Clutch is going to have it all live in the next few days. It sounds like. So
awesome.
I'm gonna head out.
You're gonna head out?
Thanks, Allie.
Thank you.
It was nice to see y'all again.
Thank you for joining us.
No
Bye, Allie.
problem. Bye.
I'll see you tomorrow, Allie.
All right. Bye.
Bye.
There we go. Okay. Back at it with the full panel.
Yeah, so for those of you that are interested in panels, that now it's up to a 30 gene panel. So it was a 22 gene panel or 22 morph panel. Now it's up to a 30. So it's going to include everything except for what we're coming out with now. So TSK, we just said, is brand new. That's not going to be on the panel because that takes time for me to optimize and get it to work in the panel. So we said after a few months, we would add the newest one. So now you will have Puzzle, you will have the Blue-Eyed Lucy complex, you will have Hurricane. All of those will be part of the panel. And since we're adding 8 more tests, we upped the price, but we only upped it $10. So it's $130. And the reason why we can do it at that price is just because you're willing to wait. So we can batch a whole bunch together. We can do a whole bunch of tests on one sample. So each individual sample, there's technician time that we need to do in the lab, which Allie, you just met, is a lot of the time it's either Sean or Allie doing that side of it. So if you have a sample come in and you're going to run 30 tests on it, then that technician time per sample is the same as if it's coming in for one Fassst test. So that's a part of why we can save a lot. And then if we batch it all together and do it on one big sequencing, one big Illumina run, that helps us save money as well. And that's why these panel tests take longer. But at the same time, it also makes them cheaper for you all. So you can get, now it'll be 30 tests you can get for $130. So that's pretty sweet.
I don't know what that breaks down to as far as like cost per test, but it's really nice. Especially if you don't know what's in there, and you may be surprised, especially with these ball pythons that have had whose ancestors have been in captivity for decades now. Could be fun. Yeah, we have a couple. Let's get to a couple questions because they're starting to build up now, and it's getting to be an intimidating list. Let's see. Spot nose. Do we plan on having a spot nose test at some point?
So this goes along with a YouTube or a Instagram question.
Yeah.
This is when the levee breaks. Is there anything available in the spider complex, such as chocolate, blackhead? So we are going to have some spider complex tests available. Spot nose and blackhead are a couple that we do have some hits for. So those of you that have been following us, you know that sometimes they work out, sometimes they don't. I was telling Sean, it's always hard to decide how much to tell people because people want to know what we're working on. But then if I tell people what we're working on and it doesn't work out, then people are frustrated that it didn't work and asking why it's taking so long. So have to kind of balance that. But yeah, we should have some spider complex. We should have some fire complex. What else? We'll have a little bit of a lot of different things. That's for sure.
Awesome. Sean,
okay.
What happened?
So I found out why Allie left. So for everybody who's interested, Allie has a tortoise. The last time she went to an NARBC show, she was talked into purchasing a little baby tortoise, and she was showing it to us and how big he's gotten. And since she bought him at NARBC. Anyway, she just texted me and said, sorry, I had to go. I was going to stay till 7, but Tony peed all over my lap.
Nice.
I'm sorry.
So that's why she ducked out. Now we have to make Tony the tortoise famous for peeing all over Allie's lap.
Then do you still have that shirt next to you?
I don't. But yeah,
man,
that's
okay.
good. That's a good tortoise destruction. That's Patrick's tortoise that tipped over the toilet. A lot of you are probably familiar with that. When we did the snake discovery episode, I guess that's been like 2 years ago now.
Almost.
At least 2, maybe 3.
Maybe. But yeah, anyway.
Tracy Sweat asked about GCR and MJ again. I know she probably missed it by 2 minutes, but it's looking like they're compatible, but we don't have enough of one of the sheds. Which one was it that we need more to really verify it?
I have more MJs, so I have lots of GCR. Thank you to Jeff and I was going to remember Jeff and Joey for sending in. I think they sent 70 GCR sheds, whether they're homozygous or heterozygous. So thank you for doing that. We got lots of those. It's Gateway City Reptiles the GCR comes from. So I got lots of GCR. The few MJs I've run so far all match, all are identical to the GCRs. So the MJs look like they are the same, but I haven't run enough. It could be those couple of people, maybe they were mixed up, and they thought they were MJs, but they're actually were not. So anyway, I do have some with lineage information from Canada that are MJs that I'll run on the next run. So in another week and a half, then I'll feel more comfortable saying for sure.
That'll be cool. From Xander, he says, do you think blackhead will be difficult since ringer and red gene seem to be hiding within the gene? Thanks. From Royal X Reptiles.
Yeah, I don't know. We'll find out soon. That's one that I want to work on. I'll be working on a pile of them over the next 6 weeks. That one I think is going to be either what I'll work on this next week or the week after. So it's coming soon.
Cool beans.
Someone was asking about Cypress. That's another spider complex. That's another one I've got a good location for.
Ooh,
so I'll be
cool.
working on that one. Yeah, I think that will be helpful for a lot of people.
Yeah. Anything on Barnhart Black Pastel?
Yeah. I actually have I have a whole bunch of shed shout out stuff to do, and part of that's Barnhart. He sent some more. Tom sent some more Barnhart sheds, some homozygous ones or super Barnhart Black Pastels. So I do have location for Barnhart, but it might take. Oh, there's Tom right there in the comments. Yeah.
He just popped.
Great question.
Yeah. So I did get
Silent but deadly
sheds, Tom. So yeah, I do have a location, and what I can see so far, which this can change, but what I can see so far, it does look like there's a different haplotype. So it looks like the variation around where the Barnhart line mutation is, or where I'm seeing it, does look different than the normal black pastel. So my guess is, like what we said, obviously it tests negative for our black pastel test right now, but it looks different. But whether I can develop a test with what I have right now, it'll take a little luck. We like to have 40 to 50 sheds if possible. If we get 20 to 40, then sometimes we can get it. We're kind of in that, range of where we might get it. But Tom sent more in, and there's a few other people that said they'll send more in. So if you have Barnhart line black pastel, especially super version, send them in and we'll get even closer.
Awesome. Hey, this is cool. Somebody, hi, Maddie. They just now discovered that we do snake genetic testing. So cool. Glad you got to find us on a live stream. So hi. So is our stuff available in Australia?
Australia, what you would need to do is figure out how to legally ship your sheds from Australia to us right now. Later, when we have the genomic registry, so there's a test that we're working on where we will do sequencing of the whole genome. There's a way to do that in sequencing centers, which we'll find a partner in Australia and other countries as well. But once we have that working, then we would be able to you could send a shed in to whoever our partner is in the country you live in and have that sequenced in your own country. And then they would just send the data to us and we would analyze the data and give you your results. So that is something we'll be able to do, but right now we can't. And it would be some legwork on your end. I think Australia of all countries would be one of the more difficult ones to talk them into letting you send sheds without, even if you were willing to fill out CITES, you're going to have to make friends with your regulators there, unfortunately. Yes.
That'd be a good one though. But I know we have partners in where are they at right now? I know we have some folks, like there's something worked out in, I believe, Thailand. There's Japan. I know there's somebody shipping out of the EU as well.
Yeah, UK.
The UK, yeah, the UK. That's it.
Yeah, UK, South Korea, and Japan, I think.
Yeah.
So the Clutch has worked out a way for people in those countries to be able to purchase tests through Clutch, and then they do the legwork for you, or have someone in those countries to help get the sheds sent in legally, and all the paperwork's done. And so you just pay for the test through Clutch, and then send that to the local person, and then they will take care of making sure that those sheds legally make it to the United States.
So if you're not in one of those countries, you can still order tests and send us the sheds, but you have to fill out the proper CITES export paperwork from your home country. If it comes to our lab and it doesn't have that paperwork, I have to take it down to Fish and Wildlife, and they either approve it and give me the shed back, or they throw it in the "we're going to burn this" pile. So you really want to make sure you have the appropriate CITES paperwork. And really, it all depends on which officer is working down there at the time. The more I have gone down there, it kind of seems like the easier it's gotten, but it would just be better if everyone follows your country's laws and fills out the appropriate CITES paperwork. And unfortunately, some countries charge a fee for that. I think Canada was like $100. It was like ridiculously expensive to submit a CITES application for a single shed. But other countries, it's free. You just have to fill out the paperwork so that you have the appropriate stuff when we get it. We have the appropriate license to receive it, but if it, we can't jeopardize our license by receiving sheds that don't have the right paperwork. So please fill out the appropriate CITES paperwork. Otherwise, you could pay for a test, send it to me, and then I don't get it because Fish and Wildlife confiscates it. So
fun stuff. I imagine that'd also be something like potentially worth talking to relevant legislators as well. Because I mean, there are things that protect protected species from being, poached or taken overseas, which is what a lot of these laws are there to prevent. But in the case of animals like this, where we're just doing genetic testing, having riders on changes to legislation could also help out. But that is your mileage may vary depending on where you're at and going forward. And this is a very new thing. So that's why we're kind of sometimes not necessarily falling in the cracks, but things are a little weird.
Right. Like we don't want I say the Fish and Wildlife, International Fish and Wildlife, I guess, doesn't want animal parts like snake skins, python skins being easily traded over, over borders. But a shed skin's different. It's a bunch of dead cells that, would have
fingernail clippings from snakes.
Right. It has no commercial value, really, I guess is the point. You can't make a set of boots out of a shed skin, but really no one's ever been in a position like ours where we want to take some of that useless material and actually use it for something. So, and it's something that really can't really harm anything. So anyway, we're working on it.
Oh, that's cool. Love from Malaysia. I've tried shipping out from my country, but it got stopped at the JFK airport. Oh man. That's cool though.
That's what I'm talking about.
Yep.
Sometimes it doesn't even make it to us. Hey, a couple people have asked about the DG reruns on the DG lit sheet. You want to talk about that a little bit, Ben?
Mm-hmm.
Yeah. So what I'm doing is I am still fitting those in. So any that are labeled as, especially if it worked for some. So like if you got either A or B or C, if you got rerun all the way across, then you're probably going to have to send in another DG shed because that shed's just not working for us. But if you have like a result for either A, B, or C, then I am going to come back through and run those. But I have to fit them in where I can. So right now, where we have a whole bunch of new research stuff, this last Illumina run, I did not do any of from the free DG sheet. Next week, when we do another Illumina run for the panels, I'll see if I can fit some more in. And it may even get to the point where we'll design a fast test for DGA and B and just run them as fast tests just to get through them. Because like we've kind of explained in this live, the Illumina, the big Illumina runs for the panels, we can fit a lot of stuff on there, but we do almost always fill them up. So if it's for the free testing, those obviously are going to only go on if the paid samples are already there. So, but yeah, we are still keeping track of that sheet, and we'll make it back through.
Good stuff. Let's say speaking of panels, wanted to quickly bring up our sale on the Blue-Eyed Lucy complex and Yellow Belly complex. Those prices have reduced. And Sean, did you want to talk about these guys?
I mean, real briefly, we've already kind of had a cheat code for you guys. We've said if you order any two from Yellow Belly or the Bell, that we would run the whole complex for you. Well, Morph Markets made it official. They went ahead and just put up the two complexes available on their own. And honestly, they've been received pretty well. I mean, you'd be surprised how many people have a Blue-Eyed Lucy and just want to know what's in it. So they'll run the whole complex just to find out what else is in there. So Yellow Belly complex is a little bit cheaper, but that's simply because it's fewer tests. Right now we're running 6 for the Bell and 5 for the Yellow Belly, right? I guess
Which
6.
ends up, we have to do 2 tests
anyway.
Yellow Belly, so yeah.
Yeah, so it's 6 also.
Yep.
Yeah. It's like technically 6. Yeah.
Which is an interesting point, actually. I keep going back to all the research that we've started doing again, that we kind of shelved for a few months. It continues to prove out that about 1 in 5 genes that we're working on turns out to have something weird about it. Either there's a second mutation that has to be identified and a separate test design for, like a Barnhart black pastel situation versus a regular black pastel, or Ultramel A, Ultramel B, or if it's, some other, deletion, like you find in, Yellow Belly, right? So it's about 1 in 5 right now is not as simple as everyone thinks it is. Yes, to the hobby, it may seem a simple recessive, but from a testing standpoint, it's actually a lot more complicated. And it's more tests than we ever thought. When Ben and I started doing this, we did not think it would be this in depth, but it actually is. There's a lot more to this than we all thought. So it's a lot of fun. And if you geek out on it like we do, then you get a kick out of it, right? But it's like, it's just, again, we found just in this last run that GCR and the other one that's escaping my brain now, Xanthics are likely going to MJ, thank you, are likely going to be, compatible.
Rich McCall Serpents, he joined on Instagram. Hey, Rich, I know that's a line he's worked with, so find that interesting. I think he had that Axanthic pop out for him in some Orange Dream Pied combo stuff back at the
interesting
back. So another Mantooth captive bred reptiles. He said, what are your experiences with longevity of actual skin? So the DNA. So I have sheds that I collected. It would have been 2016. And I had for like until 2012 or 2022. So that's what, 6 years. They were in a fairly damp basement. They were in Ziploc bags, but they're in a damp basement. And then they were in a garage it's not an insulated garage, but they were in the Ziploc bags. And I've still run those now. I just every once in a while I'll run one. And as long as you remove the shed quickly after it's been shed, it doesn't sit. If it sits in water or high humidity, that's where the damage happens. As long as you take that shed out of the cage as quick as you can and let it dry completely for a day or two, and then just get a small piece that's clean and dry and let it completely dry and then put it in a Ziploc. So that's what, 2016 till now. So that's 8 years, and they still run. So if there is a point at which they don't run anymore, if it's collected right at the beginning, if there's a point at which they don't run anymore, I don't know what that is. It's definitely over 8 years.
Mm-hmm. You were raising your hand, Sean?
Yeah, I got multiple things to say about this because I'm the person who opens all of the poopy snake bags when the sheds come.
I did a little bit of that too.
That's not
everybody's you've been hopefully taught now that water is the enemy of DNA, right? However, it's totally okay if you have a shed that's not rolled up and laid out flat. And if it's got a bunch of crap on it from the cage or maybe some, other unknown substance, to rinse it and clean it. As long as you dry it, and once it's dry, we're good. It's putting a damp. It doesn't have to be wet, but you know how a python shed. Okay, I timed out there. You know, a python shed can be kind of wet and smooshy, for lack of a better word. If you just jam that into a bag, even if part of it's dried, but that rolled up section is still wet, and then seal that bag, that moisture has nowhere to go, and that DNA is going to be toast by the time I get it.
Yep.
So if you had just ripped out that small piece that was dry and just sent that, it would be fine. But if you put it all in there and seal it in a Ziploc, yeah, the whole thing's going to be toast.
It just wrecks everything. So, yeah, I mean, you can wash your sheds, guys. I mean, it's not that you can't get it wet. It's that you don't want any moisture at all in the bag with the shed when you send it to me. So once you take it out of the cage, if it's clean, get it bone dry, send me a piece the size of a quarter or silver dollar max. It's all I need. And send it. The more shed you jam in a bag, there's more chance there's moisture trapped in there, and it's going to wreck the entire shed when I get it. So, and it's pretty predictable. I can look at a shed in a bag, and I can see the moisture inside of the bag because it didn't have anywhere to go. And I already know if I run that, it's going to be toast.
And sometimes it's green or yellow too.
Yeah.
Yes. I was at Ben's a couple of months ago, and I picked out this big fluffy sandwich bag. Just someone had taken a bull snake shed and just shoved the whole thing in there. And it was green, and I didn't open it because I knew if I did, there would be a puff of mold that would come out of it. You can also use envelopes. As long as it doesn't get wet or anything in transit, those also work. So
yes, please. Yeah. So that is the only downside. If you have everything in an envelope, you have small envelopes in one big envelope, and it gets wet during transit, then that might be a problem.
Which has also happened.
Yeah.
Yes, it does happen.
But if you have them in envelopes, your sheds are in the envelopes. The good thing about that, when it's at your house, if you're collecting them over 2 or 3 weeks and they're in that envelope, they can dry that whole time. You're not trapping the moisture in there. Mm-hmm. As long as you then put those into one big Ziploc, or you put them in some kind of mailer that's, like a plastic that you can seal fairly well, then most likely it's going to be fine. So that's the only thing you have to watch out for. And like wherever you save it, you at your house, like if you have that pile of envelopes sitting in your really high humidity snake room or something, and then that whole time they're sitting there, if you're collecting them for 3 weeks and they're in high humidity, then they're getting chewed up the whole time.
Mm-hmm. Definitely. Let's see. The questions are starting to pile up again. Eeny, meeny, miny. This one. Tri-stripe. Anything on that?
Yes. So thank you so much. People have jumped on that. I have a bunch of shed donor shout out stuff, and I've got a good one for tri-stripes. So unfortunately, they were here at the beginning. Colorado Cold Bloods sent me a pile here. So got a whole bunch that are either visual tri-stripe or het tri-stripe. So thank you, Colorado Cold Bloods. Tri-stripe. We have started sequencing it. I will most likely need more tri-stripe sheds. So if you know someone that has visual tri-stripes, send them in, and we will definitely keep at it. But it is in process.
Good stuff. Let's see. From Selective Creations, will you be able to differentiate between fire, vanilla, and disco?
I don't know for sure yet. I know that we have a very good hit or 2 or 3 really good hits for fire. We know that we have a location pretty narrowed down, and so that one I'll be able to look at. Disco and vanilla, I have some sheds for, but if you have super discos or super vanillas, that's something else. We could use a few more. My guess is it's going to be similar to the Blue-Eyed Lucy complex, and some of them we can differentiate, and some of them, are identical. So lesser and butter ended up being identical. But there are certainly, and yellow belly as well. The yellow belly complex, they're all mutations in one gene, but we can actually tell most of them apart. There's 2 different mutations that both give us yellow belly, but the others are all a little bit different visually, and we can test for all of them. So like with yellow belly, we can actually test for the 2 different mutations and tell you which one you have. If you have a -1 or a -17, but visually you can't tell the difference. So we'll see with fire, disco, and vanilla. We'll find out here before too long.
Good stuff. Oh, before we forget, in 5 minutes, those of you who are on Instagram, so we're sending this to you, or this live stream through StreamYard, but there is a limitation on Instagram where it'll only give you a live stream for an hour, and then Instagram cuts it off. So if you want to keep watching, in about 5 minutes, Instagram's going to cut out. So hop over to either Facebook or YouTube. We'll still be live there, so you can catch the rest of it. Just because we don't want to lose you. So
thank you for remembering that.
Yes.
And Rich McCall Serpents asked about Stranger. That's another one that we've started collecting and sequencing. And I don't have enough. I don't have a good location for it yet. So any of you that have Strangers, that's another good one. Someone asked about Mahogany earlier. We do have a pretty good location on Mahogany over the next 6 weeks. That's one of them that I've got a decent chance of getting. I would imagine of all of them that we have, I hope to get at least half of them. So in the next 6 weeks, I think 10 to 15 is pretty good chance we'll be able to get.
Yeah. Hi, OD. Anything with that?
Yeah. So that's also part of my shed donor shout out. So
yeah,
we've got a whole bunch, a big old thing from Wesley. Another, I think there's a good 20 or 30 in here. So we are still working on that. I had a good, it was probably like an hour-long conversation, and we did it with StreamYard and recorded it with Chris Hernan and Wesley Hatmaker. So at some point we might play parts of that or all of that, and some other things that we learned. So that's taken a little longer. We're gathering different samples and testing different things. It'll take a little time, but we are working on it, and it looks like it'll be an interesting story.
Ooh, I'm excited for that. Anything on lace? I know we got a bunch of nice white lace, or a good handful of white lace, the homozygous version of lace.
We did get some. I definitely could use more. Ooh. I had another shed donor shout out here. There we go.
Yeah. What you got?
Brian Breakus. I don't know. I'm probably saying your last name wrong, Brian. I've seen your stuff in the past. He sent both high OD and lace stuff in. So there's another shed donor shout out. This is just stuff that's come in the last couple of weeks, 2-3 weeks. So thankful for all of you for doing that. We can't do it without you. We've got a bunch of crested gecko stuff that are proven males and females when we can do sex determination for crested geckos. We've got quite a few chameleon sheds coming in. So we'll have a panther chameleon test coming soon.
Yes.
Brittany Gobble from Gobble Reptiles. She sent in stuff for calico, for monsoon, for Wookie. And then she's also sent in stuff for Hatari and Atari. So the Hatari mutation is another one that we're working on. And Dark Cloud Reptiles. Randy's sent a whole bunch for that. Like I said, this is just in the last couple of weeks. Let's see, we've got some acids from John Muller from Jam Out Reptiles. We've got a whole bunch of carpet python axanthic sheds. So I was talking earlier about axanthic species too. Here's a whole pile of carpet python axanthics.
Awesome.
Here's the Barnhart ones I was telling you about earlier. Mm-hmm. Alchemy Exotics. We've got a box full of corn snake mutations. So we've got a whole bunch here.
Oh, nice.
More corn snake stuff coming. Let's see. We got Jason Bailey. I saw people asking about mandarin earlier. Jason Bailey hooked us up with several mandarins. So we're still collecting mandarins. Don't have enough to get a location yet or develop a test, but people are jumping on it. And there's several in here from Jason Bailey. Sorry. We've got from TLB Reptiles. We've got some Super Raven and Super Chocolate. So those will be helpful.
Oh, I know we needed more Super Chocolates.
Yes. And then I wanted to, this one I want to give a special thanks. This is Kagan Andrew. So the cool thing about this, so this is a big old pile of sheds here. These are all reticmorphs. And the really cool thing about this is she went through all this work to send all these sheds, sent a really nice long letter here. But the bottom line is all of these were collected and sent in, but they actually had to get rid of all of their retics because they're moving to Florida. And so don't even have these animals anymore, but still took the time to gather sheds and send them in. And unless she moves away from Florida in the future, probably won't be able to use the test. But anyway, so other species we're definitely still getting sheds in from. And Sean's been telling me, hey, we've got to make this happen in some other species. But then I get a giant pile of data for ball python morphs, and I got to get through that first. But yeah, we're definitely still very interested in other species. And this last couple of days, there's another Colorado breeder. I wish I remembered the name, but he reached out he said, this is my yearly check to see if you've got blue tongue skink sex determination yet. I was like, no, don't. Sorry. And I looked back at the text, he's been texting for a couple of years. But I looked up the information. There has been a really good whole genome done. So I've got something to work from better than I had before. So anyway, we've got other species on the brain too. Don't worry about that. But
yes, we do.
We keep getting lots of ball python stuff to do too. So
yep. I just had inspiration for a meme. I'm gonna post that later, probably this week, just so you know.
Write it down so you don't forget.
That's what I'm doing. Sean, pick another question. I'm busy.
Okay, so it's more of like a statement. Where was the question? God, I can't find it. I lost my train of thought. They call these senior moments. My kids give me a hard time about that. Okay, yeah. So there it is. Critical constrictors. Of all the genes that you've worked on, which has been your favorite to work on? What's the one that caused you the most headaches? So I don't know what Ben's going to say, but I think I know what Ben's going to say. But it kind of rolls into what we're talking about here too. We really kind of put the brakes on a while ago because people were specifically asking us about desert ghosts, and to the point that we only had a test that worked 85% of the time, and people were like, fine, we don't care. That's how badly we want that test. And we made it available. And it's continued to be one of those things that we still do research on to this day, because there's just funny things that are going on with it. The last big panel run we did, I had a couple of people who had what appeared to be visuals, but tested as head animals, right? And so we continue to work on it. Not that we think the test isn't working. We just think there's more to it than what's going on. And we're not going to say anything about that right now. But that one not only frustrated us, but it took an enormous amount of our research funds and research time. Now that we've gotten away from it, and like I said, it rolls into what we're talking about now. Now all the excitement is back into research, and Ben is receiving the benefit of that in that all these people are starting to send sheds in now because we're really starting to turn out new tests. He's not kidding. There's probably 20 in the basket right now. We're pretty confident we'll hit at least 10, if not 12 to 15 over the next, I don't know, month and a half. And there's going to be more data coming in right behind those. While we're still working on those tests, we're going to have massive amounts of data on other stuff, right? So the excitement's there. And now that we aren't, burying ourselves in some of this stuff because the hobby's asking for it specifically so much, now the research dollars are just going to the shotgun effect. Let's send a bunch of stuff off and see what we get good, clean hits on. And we're getting a lot of the incomplete dominant stuff starting to pop. GHI, fire, a lot of that stuff that's just kind of, probably it's not a het, so it doesn't interest people as much. But what we've discovered, and I think even Justin said something about this on the last podcast that we did with him, is you find out that you end up needing some of the incomplete dominant testing, right? Because you just aren't sure and you want to know, right? And even if it's incomplete dominant, if you can prove that it is a het for something, then that animal's worth more, right? So the velocity with which the tests are coming is a direct result of you guys sending sheds. So if you have sheds for a particular mutation that you would like to see tests for, send them in. We're spending the money. I mean, hand over fist, we're spending the money, and the new tests are going to start coming out. Bang, 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 bang. So we're really close on a bunch. We've already dropped TSK today. We'll probably drop more next week at the latest. So just keep them coming. But to kind of sort of answer critical constriction question, DG was a pain in the ass. and it slowed our research down because we spent so much money on it and did so much work on it. But we've learned a lot, and that's probably going to get its own show by itself because it was such an adventure, and we learned a bunch about it, and we're still learning a few things. But now that we're past that, you're seeing the results of where the research dollars are going now. I mean, things are moving very, very quickly, both into other species and with more ball python mutations. Somebody asked about, was it leopard geckos or cresteds? I don't, it was one of the
leopard
geckos.
Leopard geckos, yeah.
As far as I know, we don't have hardly any research sheds for geckos at all yet, right?
Not very much. Some probably crested is what I have the most from. But yeah, not very many leopard geckos. So if that's something people are interested in, got to collect the sheds and send them in. I can't do anything until I have sheds. So
yeah,
I've also got an invite from. I don't want to drop any names yet because I don't know if it's okay with him if I say it, but we've got some wild caught retic opportunity to collect some samples that have GPS locations on where the animals were collected, which hopefully will help us with maybe some locality testing and retics in the future. So
that would be cool.
That's going to retire a trip that I think you're going to have to take, Ben. It's out of state, but it's closer to you than it is to me. So you may have to do a weekend drive to go collect a bunch of scale clippings.
You could send me. I'd like
handle that.
Okay. We'll send you and Allie on a world tour. You just travel all over the United States and collect samples.
I need to get some clip-ins from your snakes, guys. Let's go.
Yeah.
Hey, Leviathan.
Hi, Leviathan. They say we just sent in a shed for a new sunset combo to get tested for red stripe and yellow belly, just to be as confident as possible in the ID. Yeah.
Yeah, and Allie maybe could have told us. I don't know if that was in the run that went today or if it will likely be tomorrow. I would imagine if you already sent it in a couple days ago, it's probably either in the results today or tomorrow or the next day at the latest.
Mm-hmm.
It's all getting run within a day generally.
Yeah. While we're at it, do we want to talk about the
problem when it's a rerun or something? It's usually one day.
Yeah. While we're at it, did we want to mention the pick 4 thing that we've got going on?
Okay.
Yeah. Ben or Sean, which one do you want to talk about that?
I'll do it because I've got a couple things to say.
Yes.
So this is a 30-day only opportunity, and it is day 7 of the offering. We're going to cut it off on June 1. You have an opportunity by writing on the bag, pick 4 and writing 4 genes. If you buy 2 genes, and there's a little instruction list that, there it is, that Taylor will put up. It's a kind of a 30-day run to test the waters to see how you guys would like sort of a pick 4 or maybe in the future, possibly a pick 5 list of mutations. It would be a set price. Right now, all we're doing is saying pick 2 genes, add your other 2 onto the bag that you want us to test, along with the words pick 4, so we know what it is, and we'll run all 4 for you for the cost of those 2 genes. So if you do the math, seeing as our secondary genes are only $25 anyway, it's somewhere between $65 and $50 for the first 2 genes added, or for the first gene, and then $25 for the second. You're talking about $75 or $85, and we'll run 4 specific genes for you that you're curious about that you might have in a particular animal. Now, the only ones that we're not going to honor, so we're just asking you guys to sort of spread the word. You can't order one of our $35 tests and then a $25 test on top of that. I'm going to send those back to you and say, look, I'm sorry, but I can't do those for you because it's 4 tests. We're already kind of pushing maximum density on what it costs us to run all 4, but we're doing this for a reason. We want to see how popular it is. But the reason why I wanted to chime in is it's pretty popular. We've already started receiving sheds from people just a week out. We've already started receiving bags with those orders. But interestingly enough, and those of you that are breeders that have clutches that are still in the incubator, we've had some breeders come through and say, hey, I would like to purchase 50 of these pick 4s. I don't have the babies available right now, but I know that you guys are going to have some tests that maybe they're not available now, but they will be in the future. Can I purchase some tests now and just use these later when my neonates are popping out, when I'm starting to hatch all my babies? And the answer is yes. I've already done 2 orders. One of them is in the podcast or in the live comments, but I will not mention their name unless they want to raise their own hand. but it's pretty wise if you're a breeder and you wanted to reach out to us. You have 3 more weeks to get an idea how many tests you might need, purchase a bunch of pick 4s ahead of time, and just let me know. And okay, so Kenny jumped in. It was a little more than $18 a test, and he bought 50 pick 4s. And there's another breeder that has now done that as well. So it's not a bad deal. And it's sort of a test because in the future, one, if not both of our vendors is going to be offering a pick 5, and it's going to be a way higher price, probably $100, for a pick 5. Right now we're offering pick 4 for essentially $75 or $85 right in there. So it's a good deal. And if you've got a bunch of babies coming, you're a breeder, that's a good opportunity for you to test some multi-gene animals. Okay, that's enough from me. I'll be quiet now.
Oh, it's okay. I kind of want to take a minute to. Oh, never mind. You just clicked on that.
Yeah, I was going to mention this really fast. So, how do I get a shed that Canova has sent in tested for other genes they didn't test for? So.
Ben, no. Oh, okay. He's back now.
Sorry.
There we go. You're fine.
How could you get it tested if they already sent it in? So, Canova sent. I went and picked up sheds from him over 2 years. It's probably close to 3 years ago now. And they've sent in sheds like almost every week for a long time. So, unfortunately, the best thing you can do is just order and send in another shed. The chances of us finding one out of that giant of a pile is not super high. We can try. You can try messaging with the animal ID, the animal number from Canova, but if you don't hear back, then that probably means we tried and we're not able to find it quickly. And so it's really much faster if the next time they shed, just pay for the test, send the shed. That's going to be much quicker for you.
I get a lot of these questions through the Rare Gen customer service email. And my general response, if it's less than 6 months old, we can probably find it because it's either in the batches of sheds that I very recently sent to Ben, or I keep a couple of months of the sheds in our lab. So if it's within a few months, I can usually find it because I don't have that many to sort through. Once it's after 6 months, it sort of goes into the library of sheds. And if it doesn't have any genes that we would find useful as far as validating new tests, we'll scrap them anyway, just to conserve space. And even if it has genes that we might be saving for, it doesn't mean we'll be able to find it. And the older it is, I had one today that was from January of 2023. And they wanted me to pull that shed. And I'm like, it would be better if you probably just sent me a new shed for a couple reasons. One, we probably won't be able to find it. And B, if we do, there's a chance it's been a year and a half that it might not run well anyway. So we're going to spend a lot of time and effort digging out a shed that has a higher percentage chance of failing. Right. So just send me a new shed. So anyway.
Plus, if we're when and if we are able to get the genomic database thing going on, then that might be able to solve some of these issues from the get-go. I can totally see, like, Canova sending, here are all these tests. We're uploading this animal's DNA to the genomic database. Then when that animal's sold, I imagine they could probably just still have that data that the buyer could get later, right? So
yes, that's exactly how it will work. Yeah, we'll have that data there, and when a new test comes up, we can just run it. So
yeah, well, cool.
You bet, John. We look forward to running it for you.
Definitely. For our folks who are not ball python people, what new species are we closest to releasing that would be significant to the hobby?
We've
talked back and forth. What do you think, Sean?
It depends. The first factor is who has sent what sheds to us. And what I mean by that is as Ben pointed out earlier, we have a lot of Xanthic ball python sheds from a lot of different lines and a lot of different people. So we're proving out a lot of Xanthic. That makes it really useful to cross into other species that have Xanthic mutations because it gives us a general location to look for those things. So, I mean, you could see Xanthic tests popping out in other python species relatively quickly. It's just a matter of, can we get some sheds for those other species, Xanthic, carpet, or Xanthic blackheads, carpet pythons, things like that. As far as other just total start from scratch species, I want to do retics, but the volume of sales, according to some statistics from Morph Market, is boas. So that's really a target for us too. However, retic people are responding and sending us sheds and offering to let us come to their facilities and collect as much genetic material as we want. We're not getting a lot of boa sheds from boa keepers. We all know you boa guys are a little bit different, right? So you got to send us your sheds if you want us to develop tests. I would like to go there next because it's the second largest market next to ball pythons. But that's one of the things we just don't get a bunch of sheds on. We don't. And I've also not gotten a lot of invitations to go collect sheds or you know, some samples from belly scale clips and things like that from people's collections. So it's really directed by what you guys are sending in. If you're sending in a bunch of things that we can cross into another species, right, and using Xanthic as an example, then that's where we're probably going to go because it was easy to develop. But if you want us to break into a completely new species on mutations that are maybe not common, like a boa versus a python, you know, there's albinism in both, but they're not necessarily going to be in the same place. You know, they're different species. So send us sheds. You got to send us sheds. If you want it, you got to send it to us. We're working with what we've got, basically.
It definitely drives us a lot. I think hog noses we got a lot of sheds for. Corn snakes we've got a lot of sheds for. That makes it easier for us to move forward. It also depends on what genomic work has already been done, like as far as sequencing the whole genome at a really deep level. If someone's already done that, paid that money and done that work, that also helps make it a little easier for us. So I guess we're still kind of deciding, but I would say hognose, boa.
Hognose and corn snakes together still are not as big as the boa market.
Yeah.
According to my research. And
That's so interesting.
we get way more samples from those guys. They're more engaged than in what we're doing. So, and maybe that's partially our fault, right? We, you know, didn't go out of our way to get those guys involved as early as we probably should have, because I know there's several, you know, just Colombian boa mutations that are out there that are very popular. We just don't have the sheds. I should probably just start picking up the phone and asking people if I can come out and start taking a bunch of belly scale clips off of things.
Yeah, definitely. So if you're in the boa community, if that's really like your niche, or you know people who are into that, definitely get in there, tell them to start sending some sheds, because that would be cool. And plus, looking at it from a business perspective, we also have to make sure that our research is going in a direction that can keep us both afloat and also give us the money to research more and add on to the species that maybe aren't in the biggest market in morph market, but are still very popular and there's a huge demand for it, like corn snakes, hognose snakes, which as a colubrid person, I love it. So I'm right there with you.
Sean and I have both kept many other species too, definitely not just ball pythons and not even just pythons. So we're interested in other species also, but you know, we've definitely have to make sure that we can pay all the bills and all that. So
the dream was years ago, right, where we have a company that's large enough and has a broad enough spectrum of tests for the entire hobby that keeps all the lights on, and we can work with like really rare stuff, you know, like Fiji Island iguanas and things like that. Not in our personal collection so much, but just genetically, right? And you know, we got to get to where the business can support that first. So two things affect it. I need the genetic material from you guys, and we're going to try and go where the volume is first. But if the volume's there, but the shed response from you guys is not there, then we're going to move on to the next one. And like Ben pointed out, right now, corns and kings are right at the front because we're getting more of those than just about anything. So
makes sense.
Kayla, did your video?
Yes, it did. I'm trying to fix it right now, but in the meantime, look at me. It's got, I mean, honestly, I've had worse freeze frames in the middle of shows before, but you know, I'm going to see if I can
screen for me.
So that's okay. Well, I'm trying to fix that in the meantime. You guys.
RMS asked, are you trying for a sex test on tortoises? So one thing that, you know, you may not, I'm a genetics nerd, so I've obviously thought about this, but many other people may not. I think a lot of people that are reptile people know there are other modes of sex determination. That's not just genetic sex determination. There are things that are environmental, like temperature. So a lot of the geckos, and then also tortoises and turtles,
Crocodilians.
Yeah. So temperature is either the main factor or the only factor in some species. So since what I'm designing is a DNA test, if it's not the DNA that causes the sex, then I can't design a test for it. So unfortunately, with tortoises and turtles, crocodilians, a lot of species of lizards that have temperature-dependent sex determination, like leopard geckos, we wouldn't be able to design a sex test for them. So we can only do it. We only have a chance of being able to do it with species that are genetic sex determination. And even then, sometimes they're difficult, like green tree pythons. We've been trying for so long, and now we have new information that says their sex chromosomes are significantly different from other python species. So don't know if we're ever going to get that, even though they do seem to be genetic sex determination, but it's still complicated. But if it's temperature dependent, then I definitely can't do a DNA test. At some point in the future, we could look into doing some kind of an RNA test. So there might be something we could find that just males always are expressing, or just females are always expressing at any age. And then you could potentially design an RNA test, which would still be, something that a lab, like, we don't do any RNA at all. RNA is more of a pain in the butt to deal with, be a little bit more expensive and more of a pain, and collecting and shipping samples would have to be done more carefully. But that's a possibility for something in like tortoises and crocodilians and species where it's environmental sex determination, temperature dependent. But yeah, we'll see.
Good stuff. Oh, there we go. Love your research, guys. Can't wait to order some tests. Are you working on mahogany and tri-stripe tests from Raven?
Yeah, so I see Colorado Cold Bloods. They got back on here. I did do your shout out. I think that you were gone while I did, but I got all your sheds here. I showed we got lots of tri-stripe going on. Thank you so much. So yes, tri-stripe. We have sheds, and we've started doing the deep sequencing for tri-stripe. For mahogany, we have a location that I'm testing. So we're closer to mahogany than we are to tri-stripe. I have a chance in the next 6 weeks of getting mahogany, but it's one of those, it's kind of a 50-50 shot whether I'll get it on the first try.
Right. Anything on rainbow as well? By the way, I got my camera fixed and I'm really proud of myself, you guys.
Right on. Good job.
Critical Constrictor says, rainbow genetic tests in the future. I find it such an interesting gene, and I wonder if it's connected to the albino candy toffee gene.
That's another one. We just need more sheds. So if you know someone that has rainbow, I actually just found a couple that came in recently that were for rainbow yesterday when I was looking through sheds for new tests. So we have a handful. One of them was from Agni, that's in Europe, that did fill out CITES paperwork. She's done it, I think, 2 or 3 times now.
Yampy Yard.
Yes, Yampy.
Yep.
So she did send some rainbows from Europe. But yeah, we would need more rainbow sheds. So if you know someone, have them send those sheds. Chocolate is another one that I do have a general location for, and I will be testing over the next 6 weeks. So I have a chance at that over the next 6 weeks.
That's one of the ones in our basket right now.
Yes.
Awesome. It's exciting. Can't wait to order them. Love Suma and tri-stripe stuff. So getting my animals tested will be amazing. Yeah.
Did we have any other topics to talk about since questions have slowed down a little bit?
I had one thing that I wanted to geek out about a little bit that's like colubrid oriented.
Yeah.
Okay. So I was watching Snake Discovery the other day, and this was from their episode where they were showing off the snakes they got at Tinley most recently, and they pulled out this little guy. That is a lightning rat snake from Scott's Great Snakes. I know Scott, friends with him, and in a nutshell, they were given a future breeding pair of lightning rat snakes. And that is one of the genes that I work with. It was founded in 2016. And like, there's like, I don't know, probably less than 20 people that work with this. I'm one of them. And I actually have my first clutch of them coming in soon. So those are siblings to my boy, Mr. Lemon Bars. So, and I saw them on on snake discovery. I got so excited.
That's
And,
awesome. And that'll be
yeah.
your very first clutch to hatch, right?
Yes, my very first clutch to hatch ever. So you see my big yellow boy with his 2 girlfriends. First clutch is going to be from the Het Lightning, the black rat snake in there. And then the white-sided girl is coming later. But I'm so excited, y'all. So I wanted to geek out about that because, oh my gosh, I felt so special indirectly seeing that. So
Yeah. That's cool.
yeah.
Man. A lot more people will know about it and be interested in it now that they had it on their show.
Yeah, they're cool. You know, big
Yeah.
yellow guys, and sometimes the heads come out like pretty reds and stuff. I love weird niche things, but also they're relatable enough that, like, you know, I can say, hey, these are like the black snakes you find in your yard, but you know, he's yellow. So I like that. Anyway, yeah, that's kind of the last topic we had like planned for today. So what next?
This is a good question. And so, question for animals that are non-CITES, would paperwork still need to be done? So the difficult thing for us in the United States is every country has their own rules. And so I don't know. You would have to ask your local regulators. Just say, hey, this is the species I'm interested in sending. I'm gonna send. Make sure they understand it's shed skin. That's not worth any. It's, you know, not worth anything at all. It's not the actual skin. It's a shed skin that would normally just go in the trash. And say, hey, this is a non-CITES animal. Am I okay to do this? I would think most countries that should be fine, but I definitely don't know. And every country is different. So some it might matter, some it might not. But yeah.
That's a good question, and that was the perfect answer. It's not up to us receiving it. It's up to your export country. So.
Yeah. And we're not the ones that are going to get you in trouble. It'll be somebody else. And hopefully they would just confiscate it and that's the end of it. But every country's different. So you want to be careful.
Yep. Definitely do your research, call the, you know, whatever your Department of Wildlife Resources is, talk to them about that. Let's see. What else we got? Critical Constrictors is hopefully going to have some visual rainbows hatching out this season. That'll be exciting.
Awesome.
Hey, I wanted to talk about panels real quick.
Yeah.
So nobody's really specifically asked this, but Justin pointed something out to us that I guess they did a Patreon thing the other day, and people are curious about the differences between the panel tests and the Fassst. Okay, so sorry. People were asking the difference between the panel tests, right? You get 30 genes now instead of 22 versus our Fassst test, and why the pricing is so skewed. I mean, the panel is 130, whereas a single gene TSK is going to be $65. And that seems a little strange. There's a couple things I want to talk about with the panel. The first is when we run a panel, we're still just using the one sample, right? So we still pull one shed, put one shed in a vial, and it's how we process that shed that makes we're able to run all the genes at once, but we try to keep it as affordable as possible because you have to wait for those results. The average wait time is 3 to 5 weeks, and in busy season, it's a little bit less, but you know, should we be charging more? Yeah, we probably could, but the trade-off is, you know, with a $60 or $50 Fassst test, yes, we're using that same shed. It's the same amount of labor, and we're getting it all done for you in a day. And it actually costs us more to run the Fassst test than it does the sequencing test. So that's part of the reason is you're paying a little more for the convenience of getting a result quickly. But number two, the other thing I want to talk about with panels is that we're at 30 genes now, and we've been saying in every live cast and in multiple YouTube shows that at some point we're going to have to stop adding genes to the panel. We're pretty much at that point. What we may end up doing is refining the panel to these are the 30 most popularly ordered genes, right? Instead of continuing to add everything. We will eventually have an option, the DNA registry, for running every single gene. But the reality with that is it's a 2 month or longer wait. And the hobby just hasn't been responsive to the long wait times like that. So we want to service you guys' needs as best we can. But that's just not acceptable to us until we can find an affordable way to run it faster. The registry's, for the most part, off the table as far as just simple genetic testing. But chemically, we got to stop at a reasonable number, and that number is right around 30. And it's going to make more sense for the panel to be the more popular genes all rolled in together. That's not happening right now, but it may have to become how we run panels in the future. So just sort of a heads up. It's a chemical mixture. I tell everybody, it's like try and make a margarita that's got 4 ingredients. Now I'm going to give you 30 ingredients, and the margarita's got to taste exactly the same, have the exact same amount of alcohol, and it's got to be exactly the same. It's sort of the same thing with the panel tests. Ben's got to re-engineer these tests to make them all work well together. And every time we add a new one, it breaks 2 others. Right. So,
or 10.
So
Please no.
At some point, it's got to stop. And critical history primer cocktail. That's exactly what it is, really. And every time we screw with it, we break stuff. And the bigger the panel gets, the fewer panels run cleanly, A to Z on the first attempt. So we're at probably the maximum number. And this is not Ben's decision so much as it's me putting my foot down and going, we got to stop somewhere. 30 is a round number. Because what happens now is we run the panel, and if we get 22 out of 30, a lot of times we're going to fast test the other 8 now to try and get you results quickly. But that kills our ability to make money on that test when we have to do that. So I would rather it work really cleanly and work really well, and to service the hobby in the best way possible. That should probably be, here's our most popular tests rolled into this 30 package. And we're not there right now because we don't have to be, because we only have a few that are not as a part of the panel right now. But this time next year, we're probably going to have twice as many ball python tests, if not more. And at that point, we're going to have to pick the best 30 for a panel, or you're going to have a pick 4 or pick 5 option to choose the ones that you need. And we'll go forward from there. But I wanted to talk a little bit about that, because the pricing discrepancy is A, us trying to be fair to the hobby, but B, from a labor standpoint, it takes almost as much work for the fast testing as it does for the panel. The only real difference is Ali is an hourly rate and Ben has a much larger salary because he's a PhD. But that's really the only difference. The time intensity is about the same. So anyway, that's the long-winded explanation, but we're pretty much getting there. And I like to talk about those in the lives, because you guys will spread the word when you go out in the hobby and people are talking about this. Well, how come they do that? Well, now you know why. This is why we do what we do and why we price things the way we price them. It's not that we're trying to get over on people. It's really a matter of the sequencing stuff is just much more efficient for the money than the single gene testing is. So
if you're willing to batch a whole bunch together and wait, if you don't mind waiting a while and doing a whole bunch at one time, it's much cheaper.
Okay.
Yeah.
That was enough of that. Sorry.
No, it's okay. We got some like 3 nice questions.
But it generated some questions, I hope. So
yes.
Well, we were talking. I just want to throw something in from Instagram really quick. So Randy from Dark Cloud Reptiles.
Hi, Randy.
A little earlier while we were talking about sending sheds from other countries, he says they wouldn't know what scale clippings are. You might have a weird fingernail fetish. So I guess from another, maybe you're less likely to get caught. They just think you're sending us your fingernails. I don't know. Creative comment. Thanks, Randy.
We can always depend on Randy.
Oh, yeah.
So Trippin' Balls LLC says, if someone thought they had a new gene, new mutation, how many sheds from that animal and its offspring would it take to prove or disprove, and how much would that cost? So that's something we've talked a little bit about in the past, not very much, but we really would need you to get at least 30, if not 40 sheds from different individuals. So if you give us the same shed from the same individual, that doesn't help. It has to be different. So you really would have to breed it like 3 or 4 generations and breed it out to, as unrelated females as you could. If you had a male with that trait, breed him to a bunch of females. And it's going to cost at least $5,000, if not 10 or more, to pay the research. It all depends on how lucky we get. If we get super lucky, maybe it's only 5,000. If we get unlucky, it could be 10 times that pretty easily. So that's why it's always difficult to decide, how much resources to put into different things. But yeah, that kind of gives you an idea. If you had 30 or 40 sheds from different individuals that have that new trait, that's when it would become a possibility that we could identify it.
All right. Let's see, from Lauren Schrader. At this point in time, do you guys tend to do full panels on research sheds, or do you just go off of what we say a snake's ID is? I have no clue how practical that is, or isn't.
On research sheds, so like when I'm
We're taking their word for it.
Yeah.
When I'm looking for like rainbow sheds, yeah, if it's to design a new shed, I don't have a test that I can do on it. So I have to go off of, and that, maybe sometimes that's why we have a more difficult time with some tests, because maybe someone made some mistakes or switched something or was told something incorrect, But yeah, we definitely, if it's to do research to design a new test, I don't have a way to test it. I have to trust, and we will certainly, like, if I know I have sheds from 5 people that have worked with rainbow for the last 12 years, those are the ones I'm going to run on the very first run to make sure I have the most certain information possible. And then after those have gone through, then I might have another 20 from other people that I don't know, but they sent them in as rainbows. And then that's going to help add to it. And if something from those groups doesn't seem like it fits, we can just throw that out as an outlier, while we're designing the test. Now, once the test is designed, then we'll start testing it on known and unknown samples from people. And sometimes, like I was mentioning earlier with GCR, there's 2 that were sent in as being homozygous for GCR, but they tested as het. So now I've got to find out, there's 2 different main possibilities. One is the sheds just got mixed up either on their end or my end, or the test isn't right yet. And it's just those 2 individuals that it doesn't test properly on. And we have to figure that out. So hopefully we'll get those sheds back from those people and they'll run clean and the test is done. But if not, then we'll have to do some more work on the test. So it's definitely a very iterative process. And sometimes we'll have something that looks really good and get pretty close to the end and then find out, it's not going to work. And we got to go back and do some work back at the beginning again.
Let's see, from Josh Alden. Is it possible to test for why spider and champagne can be lethal? Super cinnamon has kinking and other genes that have problems, and use that data to see if future genes might have problems?
So that would be someone different than me. I'm a DNA code, person. That's what I really understand. That kind of a question would be more for someone that's into proteins and into like metabolomics and just different things that I don't really think about. But yeah, that's certainly possible that you could figure out mutations that change the way a protein folds or how it interacts with another protein. And you would be able to do research and see which ones are going to be more likely to cause problems. But that is definitely not my area.
Fair enough. Let's see what else we got. Selective creations. So are you able to tell if a mutation is truly recessive as opposed to an incomplete dominant? Settle the lifelong debate of paint being recessive or incomplete dominant.
So I might sound a little harsh when I answer this question. I can see Sean smiling. He's heard me answer this before.
I've had to answer it lots of times too.
Yeah. So the DNA code doesn't care whether it's recessive or incomplete dominant. It is what it is. So that those names are things that we as people give them. So the code in the animal, that DNA code, if it has a mutation, like for albino, if it has one mutation, then it's heterozygous for albino. And that's what's in the DNA code. That doesn't tell me how it looks. It just says, okay, this is heterozygous for the albino mutation. And if it has 2 copies of it, it's homozygous. And so whether it's albino, 1 copy is a het, 2 copies homozygous, or it's a pastel. If it has 1 copy, it's a het. If it has 2 copies, it's homozygous. That's all that's in the DNA code. What we see on the outside, we see, okay, I can't tell a het albino apart from a normal. So we're calling that a recessive trait. With pastel, we have a normal, we have a pastel, and then a super pastel is even more drastic than a pastel. And so we call that an incomplete dominant. But yeah, like there isn't a way to look at the DNA. That's kind of like the last question. If you're just looking at DNA code, there's absolutely no way to tell whether something is recessive or incomplete dominant, because it's just whether there's 0 copies, 1 copy, or 2 copies. That's it.
A good visual might be to bring up that photo of that gel from earlier.
Yeah.
And whether it's incomplete dominant or a recessive, to us, it looks exactly the same when we test it. It's how you guys identify it as hobbyists. Like this right here is showing 2 different examples of what a het looks like on a gel. You can visually see the difference between a negative and then the controls on the ends, right? So whether this is an incomplete dominant black pastel or a recessive albino, to us, it looks the same when we run the test. The terminology the hobby uses is what the hobby uses to describe visually how you're going to see that trait in those animals. But from a genetic standpoint, it's an identical test for us. It's either negative, homozygous, or heterozygous. That's it.
Yeah.
And to be clear, the sides with the little ball pythons over them, those are representing the homozygous. So
sorry. I thought those were controls. Okay, so those are the homozygous.
Yeah.
Yeah, but yeah, same thing. Like that could be a super enchi that has that single band that's at the top, or it could be an albino that's that single band.
So that's a perfect way to describe it. Actually, if you look at the column on the far right, that's a super enchi or a visual albino. If you look at the column to the left, that's an enchi or a het albino. Yeah. It's exactly the same to us when it comes to the test.
So yeah, so that specifically the question with paint and sentinel, unfortunately, we can't help you with that. That can rage on. People can say whether they think you can tell the difference. Now, if you're asking from like a genetics terminology, if you're looking at a genetics textbook, if you can reliably tell the difference between a heterozygous individual and a homozygous individual, then it should be called incomplete dominant. If it's not, all the time, like with pied, I've hatched out posset pieds and been able to pick out pretty good. I would say 70-80% of the time. In a lot, some combos it's impossible, but it's not all the way there. So I wouldn't call, some people could make an argument that it's incomplete dominant, but I wouldn't call pied that, and most people don't. With paint, sentinel, I can't give you an answer from a DNA testing standpoint at all. From a genetics textbook, the question would be whether you can reliably, like over 99% of the time, tell a het apart from a homozygous. If not, then I would call it recessive. But unfortunately, the DNA test can't put a stamp on that for us.
As with many questions in genetics, sometimes you ask what seems like a simple question and it becomes a whole lot of variables. So it's okay if that was a little bit bewildering, but hopefully we're going to do some episodes in the future that are like back to school oriented that could maybe explain some of the functions that could make a recessive gene look technically incomplete dominant, maybe. But let's see, from Monster Ranch
is DNA test will never answer that.
Yeah. From Monster Ranch Exotics, they asked earlier, have you noticed any differences with RK asphalt? We were talking about like black pastel versus Barnard.
I don't remember anybody sending in an RK asphalt. So
Me
we would have to have those sheds sent in, and we can test them and see if they're similar or not. If someone has sent them in, we didn't see that note that it was potentially something different. So maybe somebody has an answer to that they've tried, but we have to have the sheds. We literally can't do anything without you all helping and sending sheds. So
half of the time it's people are buying our test, sending in a shed, and when it comes back negative, then they contact us and go, hey, this is clearly a black pastel appearing animal, but your test doesn't work. And that's probably happened with, asphalt or some others as well, where people just, for whatever reason, our test doesn't work. And some people are choosing to keep that close to the chest. That makes sense,
But yeah, if whoever has that RK asphalt, if you are interested in knowing, I would happily run 3 or 4 or 5, and then do a comparison and see if we see anything different. And if it does seem to be different, like Barnhart line, we would love to get more samples and be able to develop a specific test for that. If there are enough individuals, if there's only one individual with it, then, we can't do anything yet. But yeah, that's something we're very interested in for sure.
Yeah. Okay. Lauren Schrader asked a question that I'm kind of interested in too. I'm curious to see what genes are popping up that a lot of us have no clue are even in the mix. For instance, I know a lot of folks have gotten positive cryptic tests when they weren't even looking for it. So it's fun to know. It's fun to know when we have something floating around. I guess when you're doing panel tests, have you ever gotten, is there something that you get frequently where someone's like, oh my gosh, I didn't know this was in here?
I wouldn't necessarily say I've seen a lot of any specific one, but I've seen some het albinos, I've seen some het hypos, I've seen some yellow bellies, I've seen several that were like, hey, just so you know, and some of them we traced back. I remember there's 2 in a row that were animals that people bought from Bob's Balls, and both times he's like, oh yeah, that animal was a pos het clown. I guess he's a het clown. So now not only did they find out their animal's a het clown, didn't expect it, but he could mark his male. He could change it from being a pos het clown male to being a het clown from the test that was done on its offspring, because he knew the female didn't have het clown. So anyway, yeah, we've seen that. I wouldn't say, I don't know, Sean, if any have stood out to you. I wouldn't say I've seen a cluster of anything particular.
No, yellow belly probably more than anything else, but that's just because it's such a common gene. But can you bring that question back up real quick, Kayla?
Yeah.
I actually have I've read that 4 or 5 times because I wanted to make sure I understood it 5 or 10 minutes ago before you even started talking about it. And what I initially thought she meant before I reread it half a dozen times was, are we seeing anything that a hobby doesn't know about yet? Right? Are we seeing stuff that the hobby's never seen before that could potentially be a new gene? And the answer to that is not really, kind of. How's that for an answer? So a lot of the time when we get the research back, we use software to help identify peaks that are different from a normal ball python. And some of those peaks are very common that are not mutations in the ball python hobby, but they travel with other mutations a lot. Does that make sense? I know it makes sense to Ben, but we know that DGB travels a lot with albinos also. Does that mean DGB is something cool or different? Could be. I don't know. Somebody would have to do the work. But we do see stuff that is sort of anomalous that is traveling. And if the only reason that I can think of, and Ben's the PhD, so he'll probably correct me, but the only reason that I can think of that stuff would be commonly occurring across multiple mutations, but traveling often together, especially if they're separate chromosomes, which they are, would be that there's a visual appearance that breeders are seeing in those babies, and they're breeding for that visual appearance, right? And they're unknowingly carrying a couple of other, are they mutations? I don't know, but they're unknowingly carrying a couple of other genes with that animal that make it pop more. So anyway, what's one of the interesting things about what we do is we do see some cool little quirky, what if kind of sort of things, right? But that's we can't chase those rabbit holes because we don't have time. But I mean, they do pop up.
Yeah.
Right. when we're at the point where we do have time, we want to chase them. But right now, we do see some little weird stuff like that that we can't really explain, and it would take a lot of time to go and try and acquire all of those animals and breed them out and see if there is a visual difference. But
are there any other weird?
Just from the nerdy genetic side, I think it's cool that some of these weird genes that are popping up on these peaks, and the reports that we get travel with other mutations, and they're not really close enough where they should be doing that regularly. Unless someone sees something in that animal and they make it a breeder, and they're unintentionally carrying a couple of genes along with it, which is a whole nother rabbit hole that we can probably go down in another show. But I think that's one of the really fun parts about what we do is we get to see all this cool stuff, you know, that's happening. And, you know, it's future rabbit holes, like I said, that we can chase in the future. That's what I originally thought Lauren was asking, because that's immediately where mine goes. I think that's the coolest thing about what we do is we get to see the anomalous, weird stuff. So anyway.
Cool. We're closing in on the 2-hour mark. Did we want to answer the last few questions that we have highlighted here, or
we can answer more?
Okay. Here, Ben and Sean, y'all choose. I've just been throwing stuff up there.
Looking to see if there's anything from Instagram as well.
Oh yeah, good idea.
Yeah, I think if we another 5 minutes, 5-10 minutes or so, I think that's totally fine.
Okay.
Raven asked about, have you done any research with different expressions of the same gene? Not really. The only one we did a big research kind of study on, and we're still working on that, was DG. There's clearly some weird stuff that goes on there. But besides that, we haven't gone down any of those rabbit holes either. But there are others for sure. We just haven't pursued them yet.
Someday. Let's see.
The problem with all this is it's just so expensive. If you could buy reagents for $20 and just throw crap against the wall until it sticks and you find something cool, that'd be one thing. But every time we buy reagents, it's $500. You know, so it's like it's and that's for one test. And if it fails, then it, you know, set that stuff on fire and try again. So as the costs come down, a lot more of this stuff will become a lot more feasible, but you know, it's still expensive. And when we started, 11 or 12 years ago, a full genome on a Python was $1 million. You know, when we did it a couple years ago, it came down to 25. So prices have definitely come down on a lot of that stuff, but it's still really expensive. There's so many fun projects we could be working on, but we need to be a bigger company before we start doing that. That's the stuff Ben and I really have wanted to work on since the very beginning. Once the company is doing really well on its own, we're going to be off in a corner doing mad scientist stuff.
Yes.
Yeah. Let's see. From Ixiwuna, Ben, when you were getting your PhD, did you hope or expect to be where you are now, figuring out snake morph genetics, and how difficult was it to get started?
So as a high schooler, I reached out. I've told this story a few times, but more than likely, several of you have heard it before. So as a high school, I reached out to the local university to me, and I literally just cold called their biology department and said, "Hey, is there a herpetologist I could talk to?" And they ended up giving me a number of someone, this Dr. Joe Mendelson, and he let me come in as a high schooler and see his lab and, you know, check things out. And as a grad student or as an undergrad, when I started college, I was able to do some research in his lab. But one of the things that Dr. Mendelson told me, because one of the days I was in his lab and we're in there playing with, I was doing a lot of stomach content stuff from road-killed snakes from Arizona. So they're pickled, and smells nasty in there. It's gross. We got all this goo. We got gloves on. We got this goo. And we got to take these, you know, hairs out and key what kind of a rodent it is. And it was kind of a crazy job, but that was what I liked. I enjoyed being there. And anyway, I was telling him I was complaining about, you know, stupid English class or whatever. He's like, "Ben, what you got to do is no matter what class you're in, whatever they're talking about, just think about how you can apply it to reptiles, and all of a sudden you'll be happier." So that's honestly, really what I've done. Yeah, the whole time I was in college, undergrad, and as a grad student, I was doing work on, you know, genetics and reproduction in you know, pigs and mice and cattle, all this stuff. But the whole time I was thinking about how we could use these same types of technology and tests and, you know, different things to be able to help reptile breeders. And so I had a good, you know, 6 years as a PhD student where I was learning all this cool stuff and always thinking about how I could do it for reptiles. And then just as I was, Sean and I had already been friends and talking online about carpet pythons mostly back then. But in 2011, when I got my PhD, I just finished, and Sean and I met at the Tinley show that year. And, that was 2011. So that's really, so I would say, yeah, was it difficult to get started? It just took a lot of time. So you probably have heard the story. It takes, 10 to 20 years to become an overnight sensation or overnight success. It took a long time. And Sean and I have had countless discussions over the last, I guess 2011 is 13 years.
Yeah.
We've talked about this a lot. And the very first test we finally did was in 2017. So, we started doing colubrid sex determination in the very end of 2017. So it took a long time. And then we did several years of just colubrid sex determination, where it'd be 1 or 2 this week, maybe 20 or 30 another week, and back to 5 the next week, and, very hit or miss, and just kind of kept it up so that we would, have ourselves out there as a genetic testing for reptiles. And finally it came along where we could do more fun stuff at a cheaper price. And we had the right connections, but it took a long time. So I did think about it like every single day as a grad student. And since I graduated, did I think it would work out? I was too chicken to think that it could, but Sean was always there saying, well, try this, maybe this would work. And, maybe, in your downtime, because you could tell I liked it, in my downtime, nights and weekends, play with this. And eventually it worked out.
Yeah. But I still remember that conversation because it stands out to me to this day. We were working a table together, selling our carpet pythons 13 plus years ago now. And at the time, I knew that you could do sex determination on just about every species of parrot on the planet by sending in a blood sample. Now you can do it even multiple different ways. But I asked him, how come we can't do anything like that in reptiles? And he held up his hand and he said, time and money.
Yeah.
And I said, okay, well, let's start putting it together. And that's really like, we're both hobbyists, like lifelong hobbyists since we were little kids. Like probably most of us are, honestly, right?
Mm-hmm.
But we're a really good combination of Ben's the science guy. He knows his stuff backwards and forwards, well beyond most of us lay people. And I'm a serial entrepreneur. I've owned my own businesses for 30 years now. So I'm less worried about starting new businesses than Ben was, but Ben was less worried about the science than I was. I was like, this, I don't know that we're ever going to get there. And he's like, no, we'll get there. Just, we got to keep working on it. And I'm like, okay, we'll keep doing this and doing that until we get there. And then we finally got there. So time and money, I'll never forget it. Time and money. He held up his hand and 2 fingers, just like that. And he was right. That was 13 years ago. And when we launched it, it had been 11 years, because we've been open now, a year and a half.
Yeah.
So yeah, it's a crazy story, but that's kind of the reality of most small businesses that seem like they're breakout successes. They started a long time ago with a lot of work and a lot of time. So, but it was also a lot of fun. So it doesn't suck to be able to work in something that you're passionate about and you love, right? So
for sure. Well, that was a great question. Probably a good one to end on, unless there's any other ones. That's a good one. All right.
I think that'd be a good way to bookend it. There are lots of other really good questions that I might save for maybe answering in future videos, I think.
Cool.
But yeah. Y'all got anything else?
Everyone for being here with us. We appreciate it.
Yeah, Lauren asked a question about monsoon. Lauren, go back and watch earlier. We actually answered that question in the first 20 or 30 minutes of the show.
Yeah. A lot of the questions left over are things that we have that we have answered earlier in the stream. So it'll be up on YouTube for you to rewatch whenever you'd like. I think maybe Facebook too. I'm not sure if it saves them, but yeah. Thanks guys for coming in. Thanks for those of you who have hung out for either the whole stream or most of the stream. I know have shared it around. We really appreciate you guys. And as always, give us the subscribe, comment. Even if you share the stream on your one Twitter account with 1 follower, that's okay, because it tells the algorithm that, you know, that you think that we're cool. And that's
a little bit helps.
Exactly. That's how we get people finding us in, like, for the first time in the stream, like we had earlier with Maddie. So
Yep.
yeah, thank you guys, and I guess we'll catch you next time. Thanks for tuning in. Bye.
Thanks, everybody.
Outro in 3, 2. Hang on, I'm going to turn off that ticker tape. Hang on. There we go. Beautiful. And now outro.